So when the review session is next Wednesday, so everything through Monday will be on the exam next Friday. Week and a half from now. So central dogma, transcription, translation. All the materials since last exam. Other questions? All right, while you're finishing up that Socrative exercise then, let's do a little bit of review, those of you that have already finished. It's a 3-step program. First step is admitting you've got a problem with genetics. Hi, everybody. I'm Dr. Ross. We're our support group for genetics. How can I help? Let's start with this. So there are 3 steps. If you're a eukaryote, there are 3 things that happen during transcription. The RNA polymerase makes a transcript. That's primary transcript. So what happens after that? The primary transcript is an RNA molecule that's generated by RNA polymerase. What's something that happens to that molecule of RNA while it's being transcribed? So splicing happens. That's one of the 3 steps of processing. And it's being capped and tailed. So there's capping, splicing, and 3' poly A tail is added. So 3 steps: capping, splicing, tailing. This is eukaryotes, by the way. Prokaryotes don't do this. No bacteria. We're talking about us, mosquitoes, bats. And what you wind up with is, let me draw it out in this sort of a diagram. You can have the primary transcript. That's my abbreviations, not anybody else's. Primary transcript. That's the molecule that's fresh off the presses. RNA polymerase just created that molecule of RNA. What does it look like after it's had the 5' 7-methylguanosine cap added? So down here at the 5' end, all you do is you draw 7Mg. There's a nucleotide that's added to the 5' end of the molecule that designates it in eukaryotes as a transcript. So it's a guanosine. It's like guanine. It's like the G that's in DNA and the G that's in RNA. It's just added backwards, as I said in the video. There's a 5' to 5' linkage. So that's capping. And what's the purpose of capping? It protects the messenger RNA from being degraded from the 5' end. Okay. Then there's splicing. What happens with splicing? You take out the introns. So what's an intron? Junk DNA. No, it's not junk DNA. Well, there are lots of good things that happen inside introns. So you can't call, sorry, wrong answer. You can't call introns junk DNA. There's useful things that happen with introns we're going to see a little bit later today. How do we define what an intron is?
The area of the mRNA that's not being, that doesn't code for a specific protein.
Right. So here I'm going to highlight different parts of the RNA molecule that aren't coding for amino acids. These are not parts of the RNA molecule that actually tell the ribosome to put amino acids into the protein that's encoded by this RNA molecule. These are parts of the messenger RNA that get removed, parts of the RNA that get removed before this RNA becomes messenger RNA. In other words, what happens after splicing is your transcript is shorter. The messenger RNA molecule gets physically shorter because what the cell does, the cellular machinery does, is it removes introns from that RNA molecule. Now, it doesn't necessarily, and this is what we really have to talk about today, the cell doesn't necessarily take all of the introns out every time, which is bizarre. but it's true. So introns are parts of RNA molecules that are meant to sometimes be removed before translation. So you still have a 7-methylguanosine cap. Introns have been removed. And then what's that third step? You add a 3' poly A tail. That's another enzyme entirely. And at the end of that, you have a 7-methylguanosine cap on a spliced messenger RNA, a spliced RNA molecule that now has a bunch of A's, like hundreds, usually, on the 3' end. And that is what we call a messenger RNA molecule. After it's been capped, spliced, and tailed, that's messenger RNA. It's also a mature transcript. It means the same thing. Messenger RNA molecule is a molecule that's been fully processed, all 3 steps, capped, spliced, and tailed. Sounds like something a chef would do. So that's a brief review of the video, which is a review of the reading, which is a review of transcription. Any questions now?
Well, the idea of one gene, one protein, this is what would combat that, because if you remove a certain area, you can still use the rest of it for this protein, whereas removing a different area will give you a different protein.
So it's splicing that means that the one gene, one protein rule that used to be a rule is no longer, certainly no longer a rule, is true. Splicing allows one gene to encode multiple different messenger RNA molecules that produce multiple different types of proteins. Multiple different protein sequences, in other words. Yeah?
Well, these steps happen before it leaves the nucleus, right?
That's correct. All of these steps happen before the messenger RNA leaves the nucleus. And so capping, splicing, and the addition of the 3' tail, why is it important that we know whether or not the RNA has left the nucleus yet? All of this happens that we've talked about so far is in the nucleus. Transcription and RNA processing. What happens outside of the nucleus? Translation, which is the next topic we talk about next class. Okay. So the whole primary transcript becomes a mature RNA before it leaves the nucleus and gets translated and turned into a protein. So let's see what the situation is on Socrative. Now splicing, the reason I'm focusing on splicing is because it seems to be the least well understood because, not shockingly, because it's the most dynamic part of those 3 steps. Capping's easy. Every transcript gets 7-methylguanosine added to its 5' end. Putting on the 3' tail is easy. There's an enzyme that does that. You just add a bunch of A's to the end. But understanding splicing and why the hell a cell would encode a gene that has lots of parts that get put into the RNA molecule and then all get taken out is just weird. So we focus on splicing. So, where are the consensus splice sites located? Yes, they are in the introns. So question 1B was the answer. That looks like we don't have to go over that necessarily. Any questions about that? It's the introns that contain the signals, the nucleotides that tell the cell where the intron is. The introns are the parts that get removed. The introns contain their own DNA sequences that indicate they need to be removed. By the way, what are those sequences? How do we define what an intron is?
It starts with a GA.
G
The 3'
starts,
the 3' end of the exon.
so if there's an
5'.
exon here and an intron here, And there's another exon down here. So we've got the 5' and the 3' ends of the intron labeled. What's at the 5' end of the intron? There's a G and a U, and then there's an A and a G at the 3' end. You must remember this, or have it accessible in your notes. And there's one more thing. There's an A in here somewhere. It's called the branch point, for reasons that we're not going to discuss. But that is the signature of an intron. It starts with a G, a U, it ends with an A and a G, and somewhere in between there's an A. Yeah?
You said that these splice sites located are only on the introns. Would it also be on the primary transcript too?
That is correct.
Because that's the primary transcript.
And that's why we've got people who chose choice D. It's two different meanings. So, important point. Thank you for pointing that out. So we've got primary transcript contains all of the introns. Hence, all of those GUs, A's, AGs are still there. So if we have intron 1 there and intron 2 there, each of those is going to have those sites. GU, A, AG. So they're in the introns, and introns are in the primary transcript. They haven't been taken out yet. They're not in the messenger RNA, which is why that choice was not the correct answer. So mature transcript, choice E in that case. The introns have been taken out. Or have they? Somebody argue with me. Come on.
Well, not all of them could have been taken out.
So, splicing, not every intron gets taken out. So what could you argue? Yeah, it could be E. So there could be some, what did I ask? Where are the consensus splice sites? So if there's an intron that hasn't been removed, and it stays in E, the mature transcript, the messenger RNA, then there would still be some splice sites in the mature messenger RNA. So if you put E, as long as you know that that's why you put E, that's fine. Definitely not in exons or UTRs, though. What's next? So all you need to do to answer this question is to find those sequences we just talked about. Which of those sequences have a G and a U next to each other, and then at some point there's an A, and then the 3' direction from that there's an A and a G. There's 2 of them. Which is it? B and D. There's a GU and then an A somewhere in the middle and then an AG in both B and D. So nice job for those of you that did that. All right. Ooh. This is one we're going to be talking about. I didn't even have to look at the screen to know that. No, it wasn't D. So coding strand and template strand. The answer is C, by the way. So let's talk about this. Somebody tell me a DNA sequence, 10 nucleotides. Close, yeah, 10 nucleotides, fair enough. Okay. So there's a DNA strand, that's one strand. Here's its complement. Okay, which polarity do you want to do? 5' to the left on the top? We'll do normal polarity. All right, we are going to pretend like we're polymerases. No, this is not preschool. No, I'm not going to make you do an interpretive dance of transcription yet. So there's a DNA molecule. And here's RNA polymerase. It's going to start there, and it's going to move this direction and produce an RNA molecule. Which end of the RNA molecule, so I'm going to code everything that's RNA related in blue. So as RNA polymerase moves this direction, and it winds up down here, there's going to be an RNA molecule sticking out of the active site of RNA polymerase, right? That's the RNA molecule it's synthesizing as it reads the DNA. Which end is sticking out? 5' or 3'? So all nucleic acids get synthesized in the same direction. You always add new nucleotides to the 3' end, which means the 3' end is going on in here. That's where RNA polymerase is actively putting new nucleotides onto this transcript, which means the 5' end is hanging out in the breeze. And you already knew that, even if you didn't know that. You knew that because that's where the cap gets added, the 5' cap. And that's the very first thing that happens. As soon, in eukaryotes, as soon as that 5' end leaves RNA polymerase and is in the nucleus, it gets capped. 5' end. It's the first part that gets synthesized. So RNA polymerase is actively working, adding new nucleotides down here at the 3' end of that new growing RNA molecule. Okay. So here's the weirder parts. We need to figure out which is the template strand and which is the coding strand. So we need to know which of these two strands of DNA RNA polymerase is reading. That's the template strand. And then the coding strand is the other one.
It's reading the 5' and it's coding the 3'.
Pardon? I heard two partial.
Because the RNA starts with the 5', that's going to be the template strand, because it's reading off that one.
It reads off the template strand. So is the template strand strand A or strand B? It's B. It's B. RNA polymerase reads DNA from 3' to 5'. Right, that's this orientation from 3' to 5', because it's synthesizing the RNA molecule from 5' to 3'. It's anti-parallelism all over again. In other words, if the first nucleotide that RNA polymerase is going to synthesize is the 5' end of a transcript, that's the complementary base to the 3' end of the DNA. What's the first nucleotide that's in that RNA molecule? It's complementary to the template strand. So it reads, there's the template strand, there's the coding strand. So all you need to know is RNA polymerase reads the template strand from 3' to 5' as it produces RNA starting at its 5' end. That's anti-parallel. So it's matching up as it moves along the template strand, the bottom strand. It adds an A, which is complementary to the T that's in the template strand, and then it adds, trick question, U, because RNA doesn't have T's, it's got U's. And then what's complementary to a C, G, what's complementary to a G, a C, and so forth. So what's the easy way to tell once you have the RNA sequence which one is the template strand and which one is the coding strand. The RNA sequence is identical to the coding strand, including, this is the critical part. And let me tell you, honestly, I freaked out this morning because I had to go back and review this myself. Because it's the polarity of this RNA molecule also is identical to the polarity of the coding strand. Right? It's ATGC, AUGC, they're both 5' on the left. So in all honesty, the RNA sequence and the coding strand sequence are the same, not only in the sequence of the nucleotides, but also their polarity. If they're not identical, then you're not talking about the coding strand. Okay. So let's see if we can come up with the right answer this time. Some of you had it. So there's the RNA sequence. Coding strand should be, how do you describe the coding strand? It's the same as the coding strand. Coding strand, RNA, same sequence, same polarity. So which one is the answer? Start with 5' AUG. So which one is it? C. Why is it ATG? This is DNA, it's not RNA. 5' ATG, right? So that sequence and that sequence are identical in polarity and sequence, except for T's have been replaced with U's in the RNA. So that's the definition of the coding sequence. Questions, concerns, follow-up? Clarifications. Easy way to find the coding strand sequence. Look at the RNA molecule, that's the coding strand sequence. Okay, you do something now. I've talked enough. For the moment, anyway. Practice alternative splicing. So in the video, I talked about 3 different types of alternative splicing. But I still want to know the answer to this question. If you have a primary transcript, not yet spliced, that has 3 exons and 2 introns, how many different transcripts can you create from this one messenger RNA molecule? So if you haven't worked on it yet, now's a great time. We'll take about 3 minutes. Come up with all of the different splice patterns that can be produced just with this really simple gene. Okay, 3 minutes. Go ahead. Yeah, E is exon, I is intron.
So you think if you just cut like right here?
right. This is my favorite question to ask because there are literally almost no wrong answers. So, sir, what's one splicing pattern you can generate out of this? See, there are no wrong answers. No stress. So exons are the parts of the gene that do what? They encode amino acids that are going to be part of the protein. So what's the default assumption? If I say this is the sequence of a gene, there are 3 exons, what's the splicing pattern you would predict? Do you want to keep all 3 exons, or do you just want to keep exon 1? What do you think? Do you want exons 1, 2, and 3 all together, or do you just want exon 1?
Exon 1.
Okay. So you could have exon 1 by itself. That's one possible splicing pattern. You could create that entire DNA or RNA molecule and just keep exon 1. That happens, even though there are 3 exons. What seems like the most likely pattern? To have all 3 exons together, exon 1, exon 2, and exon 3. You splice out the 2 introns. You keep all the exons. Right? To me, intuitively, that seems like that's what the cell should do. The exons are there for a reason, right? They encode amino acids that are going to be part of this protein. So what's the point of having the exons if you're going to take them out? What else do we have? Other possibilities. E1, E3. You splice out exon 2. And we're not just making stuff up. This actually happens. Yeah?
Could we just leave it the same?
You could leave it the same. It could be totally unspliced. Even though it has introns, you might go into the cell and find that you find some transcripts that haven't spliced. Exon 1, intron 1, exon 2, intron 2, exon 3. Others? Do you have tell me a third pattern we don't have up there yet.
Exon 1, well, just removing the intron 1.
Sure, just removing intron 1. So you've got exon 1, exon 2, intron 2, exon 3.
Can you just have introns?
Can you just have introns? My gut reaction is no. So, here's the way I would answer that. Introns don't get spliced together. That is, if you take out the 2 introns, which, again, would, I think, be the likeliest thing to happen, those introns get removed as structures that are bizarre. They're lariat-like. They're called lariats. This is what the introns look like after they've been removed. So they're not added together. You get intron 1, that RNA molecule, separately from intron 2. And we're not going through the details of the biochemistry of how intron 2, how those get removed by the splicing machinery, but they don't get stuck together. So you could get the 2 introns, but they would be removed. And what happens is the exons get spliced together. So no, you wouldn't get intron 1 and intron 2 stuck together. Every intron, when it gets removed by itself. Oh, here's an interesting, I think, but don't take my word for it, question. How does alternative splicing work at the molecular level? We're not going to talk about the biochemistry, but remember, every intron has that set of sequences, GU, and then an A, and then an AG. So when you have something like this, exon 1 spliced to exon 3, how might that happen?
Skips over where it's supposed to stop.
Right.
On the first one.
Essentially, so the cellular machinery, we're not going to talk about the proteins that are involved, see that GU, and then what 3' splice site do they see? Not this one. they'd go GU, oh, here's another AG down there. Splice together exon 1 and exon 3. They take out 2 introns and an exon at the same time. Does that make sense? I mean, basically, don't tell any real geneticists that I said this, but any AG, other than me, any AG potentially could be used as a 3' splice site. And that's one of the ways that alternative splicing works. Proteins in the cell say, hey, there's a GU. Oh, down there, there's an AG. Stick them together. Take out the intron. So it shouldn't actually be surprising that alternative splicing happens. Because I will not make you calculate, but you could calculate how frequently G's and U's together are on a chromosome. And the same for A's and G's, right? 1 every 16 nucleotides on average. There'll be a GU. Yeah?
One of the options that I was thinking, it was exon 1, intron 1, and then exon 3.
Yep.
But after you explain this, how would it cut it? Because you don't cut intron 1, but you're cutting.
And it turns out we're really good at this. So over a decade ago, this data is pretty old. These data are pretty old. But the percent of DNAs that are alternatively spliced, the percent of RNAs that are alternatively spliced in humans, huge. Most of our genes, almost a majority of our genes at least, and again this data is a decade old, so it's probably more than that. We're really good at alternatively splicing our genes. We do it a lot. Most of our genes, the majority of our genes, do this. One gene gets turned into multiple different transcripts that make multiple different types of proteins. Do you see any correlation between the organisms that are up here and the organisms that are down here that don't do very much alternative splicing? Seems like there's a trend, sort of. More alternative splicing on that side, less over here. Maybe not. So I just want to be able to at least show you that I'm not making all this up. That alternative splicing really happens, and it does happen. This example is fun, I think, just because we get to look at pretty fish. This is does anybody know what type of fish this is? It's got stripes. It's a zebrafish. Although there are many more types of fish that are striped than just zebrafish. Turns out zebrafish get studied a lot to understand how development happens. In this case, these scientists were interested in understanding how pigmentation, the zebrafish, happens. So here we've got a wild type fish. You notice all of these lateral stripes, hence the name. But if you have a mutation in this one gene called hagoromo, which I've been told in Japanese means dress of the gods. I'm not Japanese, so that's what I was told. Correct me if I'm wrong. When you have a mutation, what happens to the pigmentation pattern? Does it look the same to you? What's different? Yeah, the black color intensity is a little bit different, and the stripes got messed up, right? It's not stripes anymore, there's some blotchiness. So this is a gene that's involved in coloration and pigmentation in fish and maybe other species as well. And these scientists that did this study that we're talking about today are actually scientists who studied these cichlid fish from Lake Malawi. And this is an incredibly diverse group of fish. They're all really closely related to each other, but you can see, I mean, take a look. These are all really closely related fish in this poster. That is, they're species, but they're sister species. Right? So they're very similar to each other at the genetic level, but look how different they are. Some have stripes that go this way, some have stripes that go this way, different colors, different sizes of fish, different shapes of fish, sure, but the pigmentation itself, just the coloring and the striping changes depending on which species you're looking at. So what could cause all of this variation in coloration and striping? Okay, well, if you want to get back to that, then sure. Yeah, that would be a force, but at the molecular level, what actually causes, when you go inside the cells or inside the organism, what's causing these different colors and striping patterns?
Splicing patterns.
Yes, splicing. Nice work. Well, that actually hasn't been proven yet, but that was the hypothesis from these authors. Maybe alternative splicing of the hagoromo gene, this gene that is already known in zebrafish, different species, zebrafish, mutate it, it messes up the striping pattern on the fish. So this is a gene, hagoromo, that is known to be involved in striping and patterning of color in fish. So the authors, this is the authors, Itzhak Tarai and others, it was published in 2003, looked at the genomic organization of the hagoromo gene. So in this case, this gene is so large, here's a 10 kilobase scale bar, 10,000 base pairs. This is a huge gene, like 50,000, 60,000, 70,000 base pairs long. And each of the exons are these vertical lines. So look how tiny the exons are compared to the introns, first thing. These introns are huge compared to the exons. Think about this. That whole stretch of DNA is going to get turned into an RNA molecule, a transcript, and then what's the cell going to do? It's going to splice it, and it's going to take all of that back out. It's just going to leave the exons, or maybe just some of the exons. So we know this relates back directly to what we just did, what I asked you all to do, the alternative splicing, 3 exons, 2 introns, how many different splice forms could there be? That was with 3 exons, and we didn't list them all, by the way. What about 9 exons? How many different splice forms could there be? 1 by itself, 2 by itself, 3 by itself, 4 by itself, then all the combinations of 2 exons, and then all the combinations of 3 exons, and then all the combinations of 4 exons. I don't know how to do that math.
If this is what was happening to a fish, does this happen within a generation or after a week?
This splicing is happening in every fish,
So would it change
in every cell.
within the same generation?
So this is, I hope the question you're getting at, if I interpret correctly, tell me if I'm wrong, is one of the weirdest things about alternate splicing. There's variety, and it's complicated, but there's little variety within an organism or within a species. So, in other words, in every cell of a fish that's blue that has vertical stripes, so here's your horrible drawing of a blue fish with blue vertical stripes. In every cell of this organism, its hagoromo gene would be spliced exactly the same way. So the variation in splicing is not within an organism, between different cells. Every cell in a species would presumably splice the hagoromo messenger RNA in exactly the same way. But the splicing pattern would differ between species or between populations. So different species splice this gene differently. Maybe that, so this is the hypothesis, maybe that is why the different species have different color patterns. So, alternative splicing is consistent yet variable. It's consistent within an organism or within a species, but variable between species.
Is that consistency with the due to some heritability in the enzymes or other factors that might influence it?
Right, so this is the ultimate question, which we don't have a really good answer to yet, we geneticists, is how exactly is alternative splicing regulated? How does one species know to leave that intron in, but another species, that intron would get removed during splicing? Right, there must be species-specific proteins that prevent exon 1 from being removed in one species but kept in another species. So here's the punchline, and this directly relates to this thread of comments. These scientists looked in a bunch of different species of the cichlid fish, and they found that there were 9 common splicing patterns. I didn't do the math because, again, I don't know how to do that math, how many different possible splicing patterns there are, but there are 9 most common ones. These are the 9. And we get one, there's no way to predict this. You do get one that's what they call FL, full length. It's got all 9 exons, no introns, like what you maybe expect. All the introns get taken out, all the exons get kept, retained. But then there are a few of these that keep intron 2 or intron 3, but they have all the exons. They just leave in one intron. And some of them don't have all the exons. So there are a lot of these that have the first 5 exons in some order with some intron retention or not. But they all end with exon 5 and intron 5, and that's the end of the transcript. So again, splicing is variable, but it's consistent. There are many hundreds or thousands of different possible splice forms here, but there's basically 9 that actually occur in the organism in its cells. Ultimately, the authors didn't yet decide or determine whether or not the splicing of this gene does actually create the different splicing patterns and vertical stripes, horizontal stripes, and so forth between the species of cichlids. But at the very least, they found that there are some splice patterns that happen, some that don't. Yeah?
So could it be possible that other variations did come across, but were, I mean, back to the whole selected against idea, these are the ones that were able to make it, so nature says, let's continue this route. I'm sure that, they would have a baby that would have a different type of splice pattern, but it gets eaten. So that's reproduced.
Yeah, so it could be that some colors of striping patterns and color patterns don't exist in nature because the alternative splicing patterns that produced those combinations of colors and stripes weren't fit. Predator said, ooh, a checkerboard patterned fish. That's really tasty. And ate them all, and then that splicing pattern didn't exist anymore. Yeah.
And it might not be a statistically significant amount, but with all those species of fish, could some of those be due like a mutation of an intron or an exon that might change its properties so that it's one or the other?
I like the way you're going with this. So could mutation cause variation in splicing? That's an important question to ask. So how do alternative splice forms get produced? We'll get there in just a second, actually. Remind me if I don't fully address that question by the end. Okay. So in sum, processing. What you should have taken away from the video, watch it again if you didn't, please, and do come ask me questions. We've got office hours today, for example, is that cells know where to start reading DNA and where to stop reading DNA based on sequences in the DNA that tell RNA polymerase where to start and where to stop. We just looked at splicing, in which case sequences in the RNA molecule tell the cell where to splice and where not to. GU, A, AG. So it's all about DNA sequences. Okay, so quickly, back to mutations, and then you could start taking this Socrative quiz if you'd like right now, but these questions don't have anything to do with the quiz, by the way. What happens to transcription if you mutate a promoter sequence, like the -10 sequence? So let's say you had the Tada box in a eukaryote and you generated the sequence, a mutation caused it to become TGTA. What would happen to transcription? Go ahead. Yeah, come on. What sequence does Tada binding protein recognize? TATA. Do we have TATA anymore? No. So transcription doesn't occur. Right? Tada binding protein binds TATA, grabs RNA polymerase, brings it to the gene, tells it where to start transcribing. So mutation can cause changes in gene transcription. What if you mutated the consensus splice site? to GUA, there are no T's in RNA, UG. Could you splice that intron out? Not necessarily, because you've removed that DNA sequence that the cellular machinery is looking for to know that that's the end of an intron. And the same is true for a terminator. We learned in eukaryotes that there's a sequence in the RNA molecule AAU-AAA that's essential for telling transcription where to stop. If you mutate that sequence, transcription just keeps going and going and going and going and going. So because, last point, because transcription is based all on the cell knows where to start, knows where to splice, knows where to stop transcription, it's all based on DNA sequences. So mutation can have severe effects on all of these processes. Initiation of transcription, splicing, poly-A tailing, termination. So we're back here in 2 days. I will not be here. I'm going to be at a conference the rest of the week. You will be here. I've got a series of small exercises to work on. Fatima will be here. And to prepare for doing this, there's a reading for Wednesday. Do that. There's 2 videos to watch to prepare you for the exercises that we'll be doing. Make sure you bring your fully charged tablet, laptop, smartphone, etc., maybe all of them, on Wednesday. And remember, don't come here on Friday because it's Cesar Chavez Day and none of us will be here. Campus is closed. And what I'm going to do right now is I'm going to launch 2 Google Classroom assignments for you. They're not due. They're both in preparation for the next exam. So those are the details, and I hope to have the mock exam questions are due. If you're going to do them, turn them in now. I'll try to have that posted to you by Wednesday.